All right. I'm joined by Dr. Remous. Am I pronouncing it right?
Yeah, Remous.
Remous, yes. I'm joined by Dr. Remous. Please introduce yourself with what you do.
My name is Remous Ocloo. I'm originally from Ghana, and I just defended my PhD at the University of Stellenbosch. I'm a medical scientist with task applied science. We do research in clinical trials, trying to find treatment for MDR, TB, and TB. Drugs. Yeah.
Okay. You studied microbiology, right?
Yes, I did. I did microbiology.
So what made you decide to go for microbiology?
Yeah. So from let's take it from the beginning, right? I think everything that I am today was based on my scores. So when I was in primary school, which South Africans call grade school, I was getting higher scores in science. So I was drawn to things that are related to science. And then when I went to university, I was also getting very good marks in microbiology. So then I was also drawn to things that were related to microbiology. So I think that was the reason why I chose to do microbiology, because I know I will perform there, and it's reflecting in my scores as well.
Please go through your academic journey. How was it studying at your undergrad? And what is it that inspired you what is it that you did to sort of come out the other way with a degree?
Yeah, so my academic journey, unfortunately, wasn't what I wanted it to be. The reason being that I want to go to the premier university in Ghana, and I want to do medicine. But unfortunately, I couldn't make the marks. I couldn't make the cutoffs. So then I have to take a gap year and rewrite. And still, I couldn't get the marks because for some reason, I don't know, I wasn't studying enough. So I couldn't get the marks. So the only option was to go to another school, which probably wasn't the best according to the standards of Ghana. But I went to that school. And when I entered the school, I had promised myself that whatever happens, I was going to do my postgraduate outside of Ghana. So I had done all that I could to make sure that I pass, and I pass well to be able to get a scholarship to study abroad. So it wasn't easy because the campus was very limited in terms of resources. Our lecture halls were not they were not one of the best. They were not learning conducive, things were not very good. But we have to push through. And at the end of the day, I think I graduated with a second class upper, which put me ahead of my peers, and I was able to get a scholarship. So my dream came true.
Nice. So for someone who's listening to the podcast and they say, I hear Dr. Remous is from Ghana and they want to go to Ghana, right? What is it? How's the atmosphere like if we were to be comparing it between your undergrad and when you're studying now in your master's? What's the difference, and what is it that they can sort of, what advice can you give them to sort of get them ready if they want to go to Ghana?
If you want to go and study in Ghana, I think the first thing you should be aware of is that Ghana is a very peaceful country. So in terms of security, you shouldn't be scared of anything. And also, Ghanaians are very friendly. So in terms of making friends and all that, you shouldn't be scared that if you go, people might not like you. Ghanaians actually like international students or foreigners more.
Okay, sure.
As long as they can feel that you are not from there, they tend to draw towards you and love you more, show you around places, which is very good. And also, I think one thing that you would have to do is that you have to make sure your finances are covered, because Ghana, especially Accra, can be a bit expensive. So you have to make sure that all that is covered. You have your finances covered and everything. And then, you would enjoy it. And I wouldn't say which school you have to go to, because then I'll be biased. But there are a lot of wonderful schools in Ghana. University of Ghana is one of the best in Africa.
Sure.
So if you are looking into doing
maybe the top 3 for the listeners.
The top 3 schools in Ghana?
For you that you would recommend.
Okay, I would recommend University of Ghana, University of Kumasi Science and Technology, and also I'll also recommend University for Development Studies. They are also coming up now in terms of the health sciences, allied sciences. So you can, and if you're also looking on a cheaper side of things where life is more favorable, there's no too much rent and all that, you can go to the north part. That's where University for Development is. You will enjoy it. The people are very friendly. The food is very good, and the atmosphere is very welcoming. So that's my top 3.
So you get your scholarship.
Yes.
You come this side. What are the few culture shocks that you had? And maybe we can get into the academics later, but what are the culture shocks that you had, and how was it trying to adapt to a new environment?
The first culture shock I got when I came here is people are very, I don't know which word to use, to themselves. Like, it's not like Ghana where students don't close their doors. You can wake up and go into other person's room, open their fridge and take food and eat. It's that kind of atmosphere where I came from. Like, we were each other's keeper. But when I came here, it wasn't really like that. As I saw that everybody was
into themselves.
People were like in their rooms and all that. And then secondly, when I tell people I don't smoke or I don't drink, it's like, how?
You're missing out on life.
How? What do you do for fun? I'm like, no, you don't have to drink to have fun. So it's like you have to drink.
So the drinking culture is very high.
That's like the first 2 cultural shocks that I got. People think that you have to drink to have fun and all that.
So you get to start studying, doing your master's, right?
That's
when you started doing your master's. Then you went to continue on to do your PhD. Please go through what you were researching on for your master's, and just explain all the challenges and all the things, positive things that came out from that.
Okay.
During your master's.
So when I was in Ghana, I was working at Noguchi Memorial Institute for Medical Research.
Okay.
One of the renowned research institutes in Africa.
Okay.
I was an entomologist.
Okay.
So entomologist is basically somebody who takes care of insects.
Okay. Interesting.
So basically, let's say somebody who takes care of insects. So that's an entomologist. And Entomologist.
Okay,
So I was an entomologist. I was working with a company, and they were into the manufacture of mosquito nets. So my responsibility is to raise mosquitoes, like grow, let's say.
Interesting.
Raise the mosquitoes. And mosquitoes have to go from the larval stage, and then they have to go to pupae, and then now they have to then come to the mosquito itself. So it's
is that the stages of being a child? It's like
so we have the egg,
teenager, and
you have the egg of the mosquito, and then the eggs goes into larvae, and then the larvae goes into pupae, and then the pupae now goes into what? The mosquito, right? It's a very interesting process.
Okay.
Especially to see that mosquitoes are very, very delicate. They die easily. it's a very frustrating thing to do. Because how do you raise mosquitoes? But you will love it, especially when the mosquitoes hatch, and then you have to wash the eggs, and then you have to incubate it, and then they grow it, and it's a very interesting thing.
How does that look like? Because I mean, how do you wash the egg? Because I mean, the mosquitoes are already small, so that means the egg is already what do you call it? Also small. Yeah. So how are you washing it, and then how does that look like?
Yeah. So before a mosquito can lay eggs, you need to give it blood.
Blood?
Yes.
Okay.
Blood.
And then is that human blood or what?
No, normally we use animals.
Okay.
Yeah, we have an experimental unit. They have animals that are caged and monitored.
So they draw blood from the egg.
So we don't really draw the blood unless we are doing an experiment. But we have a restrainer that we use to restrain the animals, and then they keep stable. This might look a very I don't know how I'm going to say it. Sure. But yeah, we have a restrainer which is very comfortable, and then we expose the mosquitoes to the animal, and then they would take the blood directly from
them. So you do it sort of natural. So you have to go into and you release those mosquitoes into those animals.
No,
and they sort of drop the blood from.
Yeah, so it's not like release, but we would put the mosquito inside the cage where the mosquitoes are. So the mosquitoes will now naturally. So it means that the room also has to be dark because they are nocturnal feeders.
Okay.
So the room also has to be dark, and you also have to control temperature and everything. So they will feed naturally for some hours, and then when they feed, you can even see it in their abdomen.
Yes, you actually saw it. It actually shows.
It's very red, and then after a few days, they would now lay eggs, and then you just wash the egg in water, and after a day or two, you see your larvae and all that. Yeah, but it's a very interesting process. I enjoyed it.
Now, I think when you explain later, it's already sounding interesting.
Yeah, it's interesting. It's my first job, and yeah, I love it. It was so fun, especially. I was talking to you about resistance. Even in mosquitoes, you have pesticide resistance, right? So in order to be sure that our mosquito nets are going to kill mosquitoes that are in the wild, we would have to go out to take larvae from the field where like there are a lot of pesticides. And then we would come and raise those mosquitoes in the lab, and then we will now expose it to the mosquito nets, because those mosquitoes, when they become adults, they are very resistant because they were in the wild, they are exposed to a lot of chemicals, so they become very resistant. So that is what will tell you if your mosquito net is effective or not. Yeah, so going to the field, interacting with farmers, collecting larvae and all those things, it was
so larvae
nice.
It's eggs?
No, the larvae is the second stage.
Okay, sure.
Yeah, so it's eggs, the eggs into larvae or larval stage, and then we have the pupae, and then from the pupae it goes to the adult mosquito. Okay.
Yeah. All right. So I think you were still explaining.
Yeah. Yes. So I was an entomologist. So when I came here, I didn't do much of microbiology, as in culturing of bacteria. So it was difficult for me. Now you have something that very, very small. You have to culture it onto a plate. You have to incubate it. It has to grow. You have to make sure that bacteria is isolated. So we call it single colonies, so that you don't contaminate. You have to pick a single colony to work with. These things were like new techniques that I have to learn. But my friends or my colleagues, they were very, very helpful. They taught me everything that I have to know. And then because I also had a proud knowledge in DNA extraction, I harnessed on that, and then I was able to pick up quickly, and then I moved into my research. But basically, my research is about antibiotic resistance in E. coli and Klebsiella pneumonia.
Okay. Yeah. I think I only know E. coli.
Yeah. Most people
just try to explain it in a nice, friendly way so that at least the listeners can have.
Yeah. So E. coli is one of the bacteria that you would normally find in the
feces.
Yeah, feces. So it's like enteric. We call it enteric organism. So it's like stomach or gut. So it's one of the common ones that you would find.
Okay.
It can be harmful. Yeah, but when you take very good care of yourself, washing of hands and all that, you should be fine.
Okay.
Yeah. So my master's project was like a sub study of a very big project. So what that project was trying to do was that they want to see if levofloxacin can be used to prevent multi-drug resistant TB in children.
Okay.
Yeah. But then when you are exposing the children to these antibiotics, you have to be sure that the antibiotic is not causing resistance to other bacteria.
Sure.
So that is where I come in. So I picked on E. coli and Klebsiella because they are clinically significant. They cause a lot of diseases like what do you call it?
Cholera.
Urinary tract infections and all that. Yeah. So then I just collected the stool samples. We call it stool, but people call it poop. Yeah, you take the poop samples and then you have to culture it, and then you isolate the organisms.
What does culture mean?
Grow, basically.
So you collect it and you grow. How does that work?
So we in microbiology we have a selective media and we have a differential media. So a selective media will select for you. So it's like we have a particular plate. If we put Remous on this plate, Remous will have a different color. If you put Ben on this plate, Ben will have a different color. So if both of us are on that plate, they will be able to differentiate who is
between
who.
the two. Yeah, sure.
Yeah, so that's the
culturing.
Yeah, that's a differential plate. Then we have a selective plate. A selective plate is like this plate can only grow certain organisms. So probably because of the salt in it, some organisms will not grow.
Okay, sure.
Because we call like organisms that are salt loving, we call them halophilic. So those organisms will grow. But those that are not halophilic, they won't grow, right? Yeah, so that's kind of a selective. So then you can also use a plate, for example, let's say vancomycin. Vancomycin is normally for gram-positives or certain kind of bacteria, right? So it means that when you want to grow a gram-negative, you can put vancomycin in your plate so that all the gram-positives will not grow, and then what?
Okay.
Your gram-negative will grow.
Okay, sure.
Right. Yeah. So that's how you.
So you're sort of trying to control.
Yeah, trying to control basically.
Okay.
Yeah. So then you just use the stool samples. You don't have to use everything, just like a dip, and then you just put it on the
microscope.
The plate.
Okay.
You put it on the plate.
On the plate, yes.
Yeah, you put it on the plate and then you incubate because the organism has to feel like they are still in the human body before they can grow. So normally we use 37 degrees. That's the optimal body temperature.
Yes,
right?
Yes.
So we'll grow it at 37 degrees, and then overnight you will see that the organisms are there. Then you collect, and then you do the other follow-up tests.
Interesting. So you said that it's part of a bigger project.
Yeah.
But you're just specifically looking at this one.
The E. coli
and
yeah.
So what kind of support and I think maybe let me just put it, what kind of support do you sort of need as a master student in order to start and actually finish your research?
Yeah, that is
maybe to advise other people who are also
that's very good. The very first, very important, significant, and necessary advice is your supervisors. Those people are the ones who would make or unmake you.
Okay.
And most people give up on academics because of their supervisors or their mentors during their masters. So the choice of supervisor, don't just send an email to somebody and the person responds, oh, okay, I have a project for you. You can come in and do this project. I have this amount of money allocated for the project. Come and everything will be taken care of. That is very bad decision. You should actually talk to people who were previously supervised by this same person. You should actually, if you have time, if you're in the locality, if you're in the country, try and visit the department and speak to current students and see how that person is like socially. Most academicians are very intelligent, but socially, no.
They're very awkward socially.
Yes. Yeah. They are very awkward people. When it comes to emotional support, they can't give you emotional support.
I see.
And emotional support is one of the things that is needed because there's going to be a lot of breakdowns on the way. So you need somebody who can actually support you emotionally, encourage you, somebody who can motivate you. You don't need somebody who is always pushing you to give results. So I would say that the very first thing or important thing you need to do is to make sure that you have good supervisors. And then you also need an institution that has all the necessary equipment or instruments that you will need for your research. And also, I would say, has a lot of international collaborations because you don't want to be a local champion. You would want to be able to you would want to be able to visit other countries, other institutions.
Sure.
For instance, when I was during my master's studies, I didn't travel that much because I was still getting used to the atmosphere and how things work.
Trying to adapt to the new environment,
During my PhD, I don't know, I visited about 5 countries, I'm not too sure. Sure. So you need that kind of exposure. So you should also consider that a school that is like well recognized, a school that has a lot of scholarships, a lot of summer schools. Stellenbosch, for instance, you tend to go to summer schools in other countries. In America, in Europe. So you need those type of things when you want to do your master's, and you need that exposure. So that would be my advice.
Okay. That's actually very cool. I think I love the one where you talked about the supervisors. Because some supervisors, I think I also had an academic. I feel like I should have an academics podcast so that at least academics can just speak on an academic level. But besides that, I think the previous guest that I actually had he said the challenge with supervisors, sometimes they also push their own projects, which is not what you actually love or want to do. So it's very much important for look at me advising people who are masters, and I don't even have any masters. But I think it's important, like you said, that they should choose the right supervisors and do that diligence in order to be able to move forward.
I think it is true what you said, because mostly grants come with projects. So the project is already there. They just need somebody to execute it.
Oh, I see.
So if you come with your own idea and there's no money attached, you have to back that one somewhere. They will give you what they have the grant for.
100%.
Do you understand? But it's not all that bad, because most of the time when you are in the same field, the skills are almost the same. Do you understand? So that is not a
big problem.
Big problem. But the problem is that if the person cannot manage you emotionally, that is the danger. Because it's a hectic journey. Especially you are now having a feel of what research is about. You're going to encounter so many challenges, right? Especially people who travel from another province and come here. It can be a very lonely journey. You will need that support, especially from your supervisors, because they would be your contact person, right? They would be the first people you would speak to 100%. When you come to, let's say, Western Cape, for instance. They will be your first point of call, right? So they should be somebody who is easily approachable, sociable. Somebody, at least for the first 3 months, they should call, check on you. How are you doing? Like, for instance, when I came, my supervisor would tell other students to take me out. Do you understand? Just go and show him Grand West,
100%. Sure.
And then she will be like, did you go on the red bus tour? Just try and...
Just get you to feel how is it like.
Do you understand?
Sure.
So it is very important. I would say that, actually, for me, that is the most important thing when it comes to
emotional support.
Your journey. You should never compromise on supervisors, no matter the amount of money they are giving to you. No, you should make sure that they are the people that you want to work with. They are easily approachable. The chemistry is there. It's very important. Yeah.
Well, that's actually cool. So, what was the result of your master's research?
Yeah. So my master's research, what we found was that even before the children or the participants were exposed to that antibiotic, some of them were already resistant to the antibiotic. So it means that there is some kind of
intelligence.
Not intelligence, some kind of like driving force which is already causing resistance. It might be that the community is already using that antibiotic, or that something a bit on a lower level is already being used. So let's say there is a class of antibiotics, right? We have first generation, we have second generation, we have third generation, we have fourth generation. If you are already using like a first generation, which will probably be more weaker antibiotic. If you're already using a first generation, what the first generation would do is that even though the second generation is not out yet, that first generation antibiotic can select for resistance that will somehow fight against the second generation, which is about to come.
Okay.
Do you understand?
Sure.
Yeah, so that could be the reason that they might be using a lower level or a first generation.
So you sort of have to move on to fight to the fifth generation.
But how do you get to the fifth? Because it comes with research and a lot of funding. Because for the past, I think 5 to 10 years, it was very difficult to get new antibiotics. Yeah, because one thing is that nobody wants to fund antibiotic research anymore. So there was lack of funding. So the research was a bit limited. So it was very difficult to get new antibiotics to fight antibiotic resistant organisms.
Interesting. That's interesting. And this is human health, right?
Yes, it's human health.
Okay. Like you said, you are in the medical side of things as well. Okay. Interesting. That's super interesting. And I actually love understanding about antibiotics.
Yeah.
But I think I was more drawn to probiotics.
Yeah.
So what is the difference between the two, just for the sake of...
Yeah, so probiotic, actually, it helps the microorganisms or the biotics to flourish more, to be themselves.
Okay.
So we have something that we call the normal flora or the microflora. It's how originally we have been created with all the original microorganisms, because there's a lot, millions of microorganisms on our skin, in our stomach and all that, right? So a probiotic would try and rejuvenate
the normal ones.
The normal one.
Okay.
Right. So that is okay. And the normal ones, they are in such a way that they can somehow fight against the abnormal or the ones that will cause diseases. If you have a very strong microbiome or you have a very strong biotic or microbiota, it can fight when there's a disease. It can also serve as an immune system, right?
Yes.
That is good. So that is why people often tend to take probiotics so that they can rejuvenate them, their microbiota or their biotic system, if I may say that. And then the antibiotic
just removes everything.
Yeah. The antibiotic is like anti, so it's against biotic, so it kills
everything.
Yeah. So that's the difference. So actually, we should be taking more natural probiotic like yogurt and those fermented foods and all that.
The only one that I like making is sauerkraut.
I don't know
what's sauerkraut is like you take cabbage, then you ferment it.
Yeah.
So you cut it into small pieces.
Yeah.
And then you sort of press the juice out of it.
Okay.
Then when the juice comes out, so you make sure that that juice goes on top of the cabbage.
Okay.
And then you just store it in a nice cool, dry place.
Okay.
So then the fermentation process will start happening.
Yeah.
So I think I was listening to someone who was in America, but that diet is more cold. They said 7 days.
Yeah.
So with me, my fermentation took 3 days because I think this side is a bit warmer.
Yeah.
So in 3 days, it was already fermented. Okay. So you sort of put a little bit of salt just to have taste.
Yeah.
Then he says it's probably the best probiotic you can actually make because it's very beneficial. It has very nice properties as well.
Yeah. I think probiotics, I think we should all be taking in probiotics, especially. I think yogurt is the common one that we have around.
Yes.
We should be taking in more yogurt and then, to give us...
Get some plain yogurt.
Yeah, healthy...
sure.
What do you call it? Microbiota or the normal flora. Yeah.
So you start with your what do you call this? You start with your master's.
Yes.
So you're researching on the same thing. Yes. So do you sort of carry it over now in your PhD, or is it something totally different when you're studying?
I didn't actually carry over. The only things I carried over was the skills.
Okay.
I didn't actually carry over because. As I said, we have like 2 categories of bacteria or microorganisms. We have the gram negatives and then we have the gram positive. So the E. coli and then the Klebsiella species would fall under gram negatives. And then gram positive would be your Staph aureus and then your other organisms.
Is that good and bad?
I think most of these organisms are good, but as well, they're also opportunistic.
Or if they're too much, then that's when it becomes too much a problem. Yeah.
Especially when you don't take care of yourself. For instance, let's say an organism like Staphylococcus aureus, when it should be in your nose naturally, right? You can, if you swab your nose now,
a sample,
you should be able to get Staph aureus in your nose. But then if there's like, let's say a break in your nose, like a
cut, yes, or something like that,
Staph aureus can cause an infection. Yeah. So it just depends on how well you take care of yourself, and then you have to make sure that your immune system is not compromised.
Before we get to the PhD, right?
Yeah.
This is like a personal question. Do you sort of end up becoming more in tune with your body now that you're studying all this microbiome, and then instead of getting into the details of it, did it change the way you sort of see your body and how you approach your health?
Yeah, I think it does change how I take my health, especially when it comes to washing of hands. And now I tend to wash my hands more. I tend to make sure that I wash all my fruits and all that. And also make sure my environment is tidy and a lot of cleaning. The washroom is clean. And then, so I think it has kind of traumatized me a bit.
Sure.
Yeah.
Okay. That's what I wanted to know, because I feel like, because for me as an engineer, every time I go into a mall or a shop, I start seeing how the connections were done. Is it a good connection or not? And I think it's sort of the trap that we all fall into when we are doing a certain profession. You start seeing things that affect your own profession as well. So
I think it applies. It applies to microbiology.
100%.
It applies to microbiology.
So now you get to do your PhD. Please explain the journey of how PhD journey was, and how excited you are now that you're going to be.
I think luckily for me, when I finished my master's, immediately I submitted my master's. I got an email from my supervisor, telling me that they have this project. Am I interested? And I said, yes, I would like to do it. So it was not much of a hassle, but there are instances where like you'd have to search for supervisors and then all that. But when I got the project, I was a bit scared. But because, as I said, it was something totally different, because I was working on gram negatives, a different category, and then now I have to work on gram positives. Right. So I was a bit scared. And also, when they gave me like a summary of what I'm supposed to do, there is the whole project is centered around computational biology. And as at that time, I have no idea what computational biology is about and how be able to do my analysis.
100%.
So I was like, okay. But then I think as I mentioned, like colleagues, I just spoke to my colleagues and they told me, no, don't worry, you would get it, and all that. And they encouraged me. And then I took the project, and then with the help of my supervisors, I started writing my own proposal and then bringing out what I would like to achieve with the project and all that. And then, at the end of the day, we put together a very good proposal that I have to present to, once again, you need a panel.
The panel, yes.
That would have to agree that this thing you're about to do is PhD worthy. Yeah, it's a lot of work, my guy. So I presented to them, they asked questions, and then I answered, and then they had to give me the go-ahead that, okay.
How is it, the process of writing your proposal and presenting, especially the presenting in front of the panel? Is it something which you it's a scary thing, or
it's
very...
The funny thing is that even your supervisors are scared. Yes.
For you?
Yes.
Okay.
So one of the hints people give is that when you are presenting or you are defending, don't look at your supervisors.
Okay.
Because they are also scared. Don't look at them because they are also scared. Right? So it's scary. Especially my defense, my PhD defense. It's a scary thing because this is it. It's either you are getting the PhD or you are not.
Oh, you're not, sure.
Yeah. So
and this is happening proposal stage, and it's also happening in the final stage when you're actually presenting.
Yeah. So the proposal stage, that is when you and your supervisors will sit down, and then you would like put concepts together. You would write down things. Why do you have to do the project, which is very important. What would the anticipated outcome? How will this contribute to the field? What new research, what new idea, or what are you trying to achieve and all that? And then you will now organize a panel. The panel would at least have 2 experts that are in the field and people who are not in the field. So like a layman, somebody who is not familiar with the field. So they will ask, all the silly questions. So
because they're not clued up, so they will ask all as many questions.
The minor questions, things that expert might miss. So they will ask all those questions.
100%.
Yeah. So that is. So when the proposal is complete, your supervisors now say, okay, this is ready. We are confident that this would be able to
be a PhD
worthy work. Then they would put the panel together, and then now you would have to defend the work. So you will now present the work, and then they will ask you, why are you doing the work, and why do you choose to use this type of technologies or methods? You would have to defend it. Do you understand? And then when you finish that, then they will now decide. The fun part is that when you are doing a proposal defense, your supervisor can kind of come in for you when you are not able to answer certain questions, right? Because it's a proposal, you are still new. So when the panel is a bit flexible, your supervisors can come in and defend you, right? So when you are done, then they would now say, okay, they think that this work is PhD worthy, you should carry on, or maybe an expert will say that, okay, they think that if you should add this thing to it, it would make it more relevant in the community and all that. They will try to fine tune it a bit for you, and then you can proceed.
Okay.
And then now you have 3 to 4 years to finish. People who are really doing like computational stuff, purely computational, can finish their PhD in 2 years. But on average, when you are like a lab based, you have to do lab work and 3 to 4 years.
3 to 4 years, sure.
And then after that, now you have to defend. Right? This one, you're on your own. Your supervisor doesn't answer any questions. You would have to defend the work. They ask you all the questions.
Sure.
Now, when you are done with the questions, you have to excuse yourself from the room. And then another deliberation again. They will decide whether you are knowledgeable enough about the field.
Okay.
What you have discovered or what your findings are about. Do you know more? Do you have more knowledge on it?
100%.
And then after that, they can now confer the degree on you or not. Yeah, so that's basically how the PhD
process works. Okay, I think maybe, like you said, you were saying you were doing the computational biology, right?
Yeah.
And you were doing on the positive side of... just explain what you were doing so that at least when you just sort of try to close it from there.
Yeah, so the aim is to try and understand the antibiotic resistant genes that are causing the antibiotic resistance, right? Sure. Now, there are several approaches that you can use. So you can target the genes one by one, so that we call something polymerase chain reaction.
Okay.
So what it means is that you like, there is a machine, and then you design like a target. So that target would go and bind to whatever you ask it to bind. So I have it's like a magnet, right? So I'm telling this magnet that when you go into that bacteria, if that particular gene is there, bind to it.
Bind into it, okay.
So if it binds to it, it kind of amplifies. And then you will see it.
Sure.
You do like a gel.
Sort of a sample of some sort.
Yeah, you see it. So like a band, you see it. So you know, okay, that gene is there. Okay, that's like a more conventional approach.
Okay.
Then there's a computational approach where you will sequence the DNA of the bacteria, right? And then you will use a computer. Already, I know the sequence of the gene I'm looking for. So I will take that sequence and I will shoot it into the genome of the bacteria.
Yeah, sure.
You say blast. So you blast it.
Okay.
So if that sequence is in the genome of the bacteria, it will be highlighted or it will be flagged.
Okay.
Then you will know that, okay, that gene is in the bacteria.
Sure.
So that is one of the things I want to do. Right? And the second is that I want to see how similar are the organisms that I have, because I have organisms from different settings, right? So from setting 1, setting 2, setting 3. How similar are these organisms?
Sure.
So what I have to do is that first I have to sequence the DNA, and then I have to compare the DNA from setting 1, setting 2, setting 3, and see what is the similarity. Is there a transmission between setting 1, setting 2, setting 3? Yeah, so that is what I use my computational biology knowledge to achieve.
Interesting. This is really interesting stuff. I really love what you guys are doing for us. At least you're making sure that we have more class 5 resistant antibiotics.
Yeah.
So yeah, thank you so much, Doc. Why did I forget this question now? I actually had a question that I wanted to ask you.
Oh, it's fine. You can.
Actually, I hope I don't remember this question and I'm already closing. But thank you so much, Doc, for coming through and sharing your knowledge. It's really encouraging because I'm sure Elijah did mention that this is a podcast for professionals to share their journey and how it was.
Yeah.
And now that I remembered my question, what is the next step for you now?
Yeah, so the next step for me now, I'm trying to establish a next generation sequencing facility where I'm working currently, because the next wave, even in engineering, is going to be artificial intelligence. So we have to make sure that already we are moving with the waves, right? So what I want to do now is to establish a next generation sequence facility where we can sequence bacterial DNA. And then with this technology, we will be able to understand the bacteria more. You would be able to understand how the bacteria is able to cause infection, what is changing in the bacteria, especially, let's say, cancer, for instance. Cancer is due to like mutations in the DNA, right?
Sure.
But what mutations? Do you understand? So if you do like sequencing of these cells, you will be able to know that, okay, I have a healthy cell, I have a cancer cell. If I compare the two, I see that there's a change here. There's an A in the cancer cell, and there is no A in the what? The normal cell. So yes, probably this A might be the reason why this one is cancerous. Do you understand?
Sure.
Yeah. Then we can have a way of trying to tackle it. Do you understand? So this is what I want to achieve with antibiotic resistance, for instance. And then another thing is to try and come with a more effective approach. Because what I want to do is that there is like TB in South Africa, very common, right?
Sure.
But when people are receiving TB treatment, how do you know that the treatment is working? Especially, let's say, if it's a new drug that they are trying out, how do you know it's working? Okay. So what I want to do now is that I want to come up with a method that will be able to detect live TB in
the human body.
Yeah, human body. To know that, okay, week 0, the number of live TB that you have was probably 20 CFUs. But in week 2, it's down to what? 10.
So it's almost like the science of HIV and CD4 counts and all
that. And all that. Yeah. Okay. So yeah, it's going to be that type of thing. So like to quantify the live TB, because the technologies that are available right now, they count DNA. And DNA, you can count DNA of a dead TB because DNA is very stable. Do you understand? So even if the TB is dead, the DNA will still be there. So if you are counting the DNA, then you are actually counting dead TB as well.
Yeah, sure,
but I want to count a live TB. So I need a differentiation, something that will specifically tell me that this is what a live TB that I'm counting. So that's the new thing.
I hope you have put a patent on this so that they don't take your ideas.
Oh no. We'll see how it goes. It's an approach. There's already research on that.
Oh, okay.
People have already done
started working on
similar things that I will have to learn from, then I will try and use those ideas that on the knowledge from there to build something new, something that can be automated, basically.
100%.
Yeah.
Congratulations, Doc.
Thank you very much.
When is your graduation?
My graduation will be in December, hopefully.
In December, okay. Okay, I think it's going to be in December.
Yeah.
And we want to say congratulations there.
Thank you very much.
You are doing justice to the world. And I mean, some of these things we just see ourselves taking part of those, but we don't know the level of research that has went through that.
Yeah.
And when I say I appreciate you, and thank you so much for contributing positively to our country.
Yeah, thank you very much for inviting me to your podcast.
And I just want to give a shout out to everyone who's watching from Ghana.
Yeah, to my fellow Ghanaians.
Sure.
Thank you for joining. And yeah, this is one of the best podcasts in South Africa.
Come on.
And I think you should engage with us more often. And you wouldn't regret it. It's very educative. It's one of the educative ones out there. It's not all about glitz and glam. We also have to rejuvenate and energize our mind and intellectual property a bit. So yeah, stick with us.
Awesome. Thank you very much. Like and subscribe, everyone from Ghana. We just want to see those likes and those views from that side.
Thank you.
Thank you so much, Doc, and congratulations.
Thank you very much.
Awesome. Thank you so much for listening to this episode. I appreciate you taking time and tuning in and be part of our community. We get to share ideas, stories, and perspectives through this podcast. I hope you enjoyed it and found something valuable in it. Don't forget to subscribe and share with your family and friends. I look forward to connecting with you in the next episode. Until then, take care. This is Stega Podcast signing out. See you next time.